Hello, and thank you for tuning in to our virtual presentation of Homophily and Heterogeneity in the Cancerous Cell Line Network for Gene Therapy Improvements. My name is Jane Adams from the University of Vermont Complex Systems Center, and I am presenting on behalf of our international collaborative group, which includes Célestin Coquide of the UTNAM Institute, University of Bourgogne-Franche-Comté, Diana Garcia of the National Institute of Genomics Medicine in Mexico City, and Rodrigo Migueles of the Quantitative Life Sciences Interfaculty Program at McGill University in Montreal. Cancerous cells are widely diverse. Heterogeneity is observed at the individual level, at the tissue of origin level, and even within the same tumor. Seeking therapeutic specificity is therefore one of the biggest challenges of current cancer treatments. Although cancer cells bear alterations that confer them malignant phenotypes, these key alterations can also be used to target cancer cells with increased specificity. Here, we use networks to make a systematic comparison of the key genes responsible for these alterations across different cancer cell lines, looking for new potential treatments based on drug repurposing and combinatory therapy. The Project Achilles DepMap dataset evaluates the effect of individual gene depletion of 18,000 different genes on the survival of 625 different cancer cell lines. This dataset was generated using interferent RNA and CRISPR-Cas technology to knock down each gene and evaluate its effect. This information is then used to calculate gene dependence probabilities. For each cell line, we selected genes with a dependence probability above 0.5, a threshold suggested by the Project Achilles researchers. We ranked these genes from greatest to smallest knockdown effect. We then calculate the Spearman rank correlation between each pair of cancer cell lines to build a weighted undirected network. This is orthogonal to more traditional research methods such as weighted gene correlation network analysis, WGCNA, in which correlations are computed between pairs of genes. Here, we are instead comparing correlation between all pairs of cell lines. Using these computed gene dependence correlations, we first observe heterogeneous similarity within cancer types. Here, we show gene dependence rank correlation matrices among cell lines from the DepMap dataset within the same cancer type. The correlation between cell lines of the same cancer type varies and may be partially explained by cancer subtype. However, we were also interested in comparing gene dependence correlation among cell lines of different cancer types. After computing the gene dependence rank correlation matrix for all cell lines, we generate a network graph in which nodes are cell lines and edges are weighted by gene dependence rank correlation. We apply a backbone thresholding method to preserve edges which are most statistically unexpected. In order to identify unusual connections in this network, we sparsify the matrix using a backbone method previously described by Serrano et al. Unlike universal thresholding sparsification methods, this method is locally aware. It preserves edges based on the average edge weights associated with each node and keeps edges that are most unusual within the local network context. Using an alpha value of 0.032 resulted in a network with 15 components that displays 126 cancer cell lines and 186 edges. Here, we show a diagram of the backbone thresholding method from Serrano et al. Applied to our gene dependence correlation network. The backbone method works by analyzing each node within the network and preserving only edges which are statistically significant according to an alpha threshold. This threshold is chosen so as to achieve a high number of components in the network while keeping some fraction of the network links. The red line, indicating the number of connected components, begins at 0 as no nodes are connected, reaches a peak at 20 connected components, and then returns to 0 as all nodes become connected. We chose a threshold alpha value of 0.0323, where 0.095% of links are preserved and the number of components is 15. The resultant backbone network visualization of cell line gene dependence correlation reveals interesting homophily and heterogeneity among cancer types. Here, the network is visualized using a force-directed layout weighted by gene dependence correlation. To improve our understanding of heterogeneity within cell lines of certain cancer types, we also visualize the network with the addition of cancer subtype. Examining specific cell line clusters at the primary and subtype disease levels, additional nuance can be found in the affinity behavior of cancer subtype. We are particularly interested in unexpected similarities among cell lines of different cancer types as a potential exploratory avenue for combinatory therapies and for repurposing of existing therapies. Certain cancer types are more commonly linked to cell lines of a different cancer type, while others seem predominantly homophilic. For each cancer type, the number of nodes in the network graph and the homophily score of each cancer type are shown here. Of note, lung cancer and pancreatic cancer cell lines, both highly represented cancer types, are more frequently connected to cell lines of different cancer types, as evidenced by negative homophily scores, indicating that gene dependence probability rankings among lung cancer and pancreatic cancer cell lines most resemble the gene dependence rankings of other cancer types. Conversely, leukemia, another well-represented cell line, has a homophily score close to 1, indicating that gene dependence probability rankings among leukemia cell lines most resemble the rankings of other leukemia cell lines. These similarities provide an interesting exploratory analysis of gene dependence rankings. Finding gene dependence probability rank correlations between cancer cell lines of different types may help to identify similarities in key alterations, thus providing hints for combinatorial therapies and novel applications for existing therapeutic strategies. Thank you so much for your interest in this project. If you would like to reach out to discuss further, please send us a message on the NetSci 2020 or Society of Young Network Scientists Slack channels. We're very interested to hear your thoughts and ideas and answer any questions you may have. Have a wonderful NetSci!